So our mitochondria are constantly producing free radicals and reactive oxygen species. And the way this is happening is we know our mitochondria have these electron transport chains. So what happens is through metabolic processes like glycolysis and the Krebs cycle, we produce these reduced cofactors. And we know these reduced cofactors donate electrons to this electron transport chain. For example, NADH donates electrons to complex 1, which donates electrons to ubiquinone, which eventually will donate these electrons to complex 4, which donates those electrons to oxygen to form water. And the same thing with FADH2. They donate electrons to complex 2, which eventually donates those electrons to complex 4, which donates those electrons to oxygen to form water. So this is normally the way things are supposed to happen. However, every now and then, whether you're young, old, healthy, or sick, our mitochondria and our electron transport chains are constantly making mistakes, where instead of complex 1 donating those electrons to ubiquinone, complex 1 makes a mistake and instead donates those electrons to oxygen to form this superoxide free radical. And the same thing is going on with complex 3. But the point is, every now and then our electron transport chains make mistakes and prematurely donates those electrons to oxygen to form this superoxide free radical. And we know this is a free radical because it has this unpaired electron. And this unpaired electron is extremely reactive. It's very unstable and extremely reactive. So therefore, it's going to react with, modify, and destroy biomolecules like proteins and lipids and nucleic acids. So this is bad. These free radicals are bad and they're reactive and they're dangerous. They're cytotoxic. However, fortunately, we have enzymes like superoxide dismutase, which takes this superoxide radical and converts it into hydrogen peroxide. Now, once we form this hydrogen peroxide, we have another enzyme called peroxidase, which takes this hydrogen peroxide and converts it into water and oxygen gas. So yeah, even though our mitochondria and electron transport chain is constantly producing these superoxide free radicals, fortunately, we have enzymes that can eventually convert it into water and oxygen, which are safe and neutral, and they're not cytotoxic. So this is good. It's good we have these enzyme systems. However, under certain contexts, for example, if we have weak upregulation of peroxidase or just dysregulation of peroxidase, instead we can go through this pathway where hydrogen peroxide instead reacts with iron and other free metals to form hydroxide and this hydroxyl radical. And in fact, this hydroxyl radical is perhaps the single most cytotoxic biomolecule our cells produce. Because again, this hydroxyl radical has an unpaired electron. So it's super reactive and it's going to react with, modify, and damage biomolecules in the cell. Another way our cells produce reactive oxygen species is through the immune system. So the immune system phagocytosizes bacteria. So once the bacteria is phagocytosized, we process it. So the way this happens, and to illustrate this, I'm going to draw this phagosome a bit larger so we can illustrate what happens. Essentially, this phagosome has an enzyme called NADPH oxidase. And essentially what happens is this NADPH reduced cofactor donates electrons to NADPH oxidase, which donates electrons to oxygen to form this superoxide free radical. Now, once the superoxide free radical is produced, it reacts with superoxide dismutase to form this hydrogen peroxide. Now, once hydrogen peroxide is produced, we know it can react with iron to form hydroxide in this hydroxyl radical. So in this context, our cells and our immune system is intentionally producing these reactive oxygen species. And not just that, the phagosome goes a step further, where we also take this hydrogen peroxide to form hypochlorite, which is literally bleach. This is literally the active ingredient in bleach. So why on earth are our immune cells producing reactive oxygen species and bleach? Well, these molecules are extremely reactive, so they're going to react with, modify, and damage the proteins and lipids that make up this bacteria. These molecules are so reactive that they're going to react with and essentially disintegrate this bacteria, and it's going to kill this bacteria. So this is actually pretty neat. This is pretty cool. This is a way our cells essentially produce bleach to kill bacteria. So this is referred to as an oxidative burst, and this is what happens during inflammation. And again, this is one reason why too much inflammation is bad, because during this inflammation process, we produce these reactive oxygen species, which can diffuse out of the cell and damage biomolecules. So there's collateral damage. Another free radical our cells produce is nitric oxide. For example, in the endothelial cells, they take L-arginine, react it with nitric oxide synthase to form this nitric oxide, which is a free radical. It has an unpaired electron. So then this nitric oxide can diffuse into smooth muscle. And then once it enters the smooth muscle, it can react with enzymes to cause a cellular effect. So this is another situation where our cells are intentionally producing free radicals. However, this nitric oxide does not only play a functional role, it can also have a pathological role. For example, in the process of this superoxide radical, it can form this peroxynitrite, which is an extremely toxic molecule because this peroxynitrite can decompose and break down into other free radicals. For example, this hydroxyl radical and also other free radicals. And all these free radicals have these unpaired electrons. So all these free radicals are very reactive. They're going to react with, modify, and destroy biomolecules. So this is bad. And of course, under inflammatory conditions, we're going to have the superoxide radical. So that's when we're going to produce these toxic byproducts. Another example is in the smooth endoplasmic reticulum in the cells. So sometimes we have an unfolded protein that we need to fold into the proper conformation. However, to fold certain proteins, we need to oxidize them. We need to take these reduced thiol groups and oxidize them so we can fold the protein into the proper conformation. However, for this unfolded protein to get oxidized, something must get reduced. So what is getting reduced? What is gaining the electrons? Well, in this situation, it's this PDI enzyme. And the details aren't really important. The point is this PDI enzyme gains the electrons. And then once it gains the electrons, then it donates those electrons to oxygen to form this peroxide, this hydrogen peroxide. And once we form this hydrogen peroxide, we know it can break down into the hydroxyl radical and produce these reactive oxygen species. So the point is sometimes we need to fold proteins, but in order to fold these proteins, they need to get oxidized. So therefore, for this protein to get oxidized, it loses electrons. Where do those electrons go? First, they're donated to this PDI, which then donates the electrons to oxygen to form this hydrogen peroxide, which breaks down into reactive oxygen species. Also, in the context of hyperglycemia, so if we have a lot of glucose in our blood, and also if we consume a lot of refined carbohydrates, these carbohydrates in excess can have a toxic effect in the cytosol and in the extracellular matrix. Because what happens is these carbohydrates can react with proteins. And what happens is the carbohydrates first open up, and then once they open up, they have this reactive aldehyde group that can react with nitrogens in proteins. So then when they react, they essentially form this Schiff base, which then converts into this amidori product, which then forms this 2,3-enediol. So then once we form this 2,3-enediol, it can react with oxygen. And when these two compounds react, we produce two toxic products. One, we produce these advanced glycated end products, which are essentially just proteins that are glycosylated. And we also produce this hydrogen peroxide, which is again, can break down into the hydroxyl radical. So we know this is toxic, this produces a free radical, but these advanced glycated end products are also toxic because we happen to have immune cells that have receptors that can sense these advanced glycated end products. And when these immune cells sense these advanced glycated end products, they trigger inflammatory pathways and trigger inflammation. So the point is high glucose levels, and if you consume a lot of refined carbohydrates, those carbohydrates react with proteins, then go through this process to form these advanced glycated end products, which produce inflammation, which is bad. And there are many other ways our cells produce free radicals. For example, when we exercise, our mitochondria produce reactive oxygen species as a signaling molecule. Our peroxisomes also intentionally produce hydrogen peroxide. Also, when we process xenobiotics and drugs, when we metabolize these drugs, sometimes we produce these free radical intermediates. And also, if we have certain metals like iron and copper, they can also react with hydrogen peroxide to form this toxic hydroxyl radical. Also, in the presence of high energy photons, for example, ionizing electromagnetic radiation like X-rays and gamma rays and UV light, they're made of these high energy photons. And these high energy photons have enough energy to break bonds. And when these bonds are broken, we produce these free radicals. And also pollutants in general and toxins, for example, smoking cigarettes, have toxins that also produce these reactive oxygen species. And also just normal metabolism. For example, when we degrade these purine nucleic acids, we also produce these reactive oxygen species. So normal physiology is constantly producing free radicals. And you might wonder, so what? Why are free radicals like this hydroxyl radical so damaging? Well, we already explained how they have these unpaired electrons that are super reactive, and they can react with, modify, and damage important biomolecules. For example, DNA. This unpaired electron can react with and break bonds. It can break these covalent bonds. So then once these covalent bonds are broken, we have enzymes that fix the DNA. But when the enzymes fix the DNA, they accidentally make mistakes that lead to mutations that lead to faulty proteins. And also, these reactive oxygen species can also chemically react with and modify these nitrogenous bases. And once these nitrogenous bases are chemically modified, they function differently. So now when this DNA replicates and base pairs with another nucleic acid, it's not going to form a proper base pair because it's chemically modified. So that leads to new mutations, which again can be toxic and can lead to cancer. And also these reactive oxygen species can also react with proteins. For example, it can react with these thiol groups to form these new chemical modifications. So now once these proteins are chemically modified, now they're different. They're essentially different proteins, so they're going to function differently. And they can also react with these thiol groups to form covalent bonds and these cross-linking that is not supposed to happen, so now this protein will function differently. Also keep in mind that our cells have this plasma membrane, and this plasma membrane is made out of phospholipids that have this hydrophobic region, which creates this hydrophobic barrier, which acts as a barrier for the cell. So it's important to maintain this plasma membrane barrier. However, reactive oxygen species can react with these hydrophobic regions, chemically modifying and essentially compromising the integrity of the plasma membrane. So how does this work? It's a complex process, but imagine this is a fatty acid that's part of a phospholipid. What happens is this hydroxyl radical rips off this hydrogen, forming this free radical lipid, which then can react with oxygen, forming this peroxy radical, which can then decompose into carcinogenic compounds. But not just that, this peroxy radical can react with another phospholipid fatty acid, forming another free radical, and also forming this compound, which then can react with iron to form this free radical, which can react with another phospholipid, forming another radical, and this product, which can then break down into another carcinogen. So you can see how this process can get out of control very quickly, because just in one process, we also produce two other radicals, which also will go through this entire process over again. And notice these are phospholipids that are all in close proximity. So they're going to react with other fatty acids in close proximity. So they're going to constantly go through the cycle over and over again, forming these carcinogenic byproducts. So we can see how free radicals damage the phospholipids. And also they form these polar products. So these polar phospholipids essentially compromise the integrity of the plasma membrane. But it's not just free radicals that can cause this process and trigger this chain reaction. It's also frying food. Frying fats also form these toxic compounds. However, fortunately, we have a defense system against these reactive oxygen species and free radicals. So what is the defense system our cells have evolved through time? Well, remember glycolysis, where we take glucose molecules and we do a bunch of chemical reactions, eventually forming pyruvate molecules? Well, we can take this particular intermediate of glycolysis to enter a pathway referred to as the pentose phosphate pathway. And this pentose phosphate pathway produces these reduced NADPH cofactors. And these reduced NADPH cofactors can donate electrons to this oxidized glutathione to form this reduced glutathione. And what's neat is that this reduced glutathione can act as an endogenous antioxidant. It essentially has electrons that it can donate to reactive oxygen species, that it can donate to unpaired electrons. For example, if we have this reactive oxygen species with an unpaired electron, we know it's super reactive because, again, it has an unpaired electron. But fortunately, we have this glutathione, this reduced glutathione that has electrons that it can donate to this reactive oxygen species, essentially neutralizing it. So this is pretty neat. We have this endogenous antioxidant system. And the way it works is this glucose has electrons. So these electrons from this glucose can eventually be donated to NADPH, which can donate that electron to glutathione, which then can donate that electron to reactive oxygen species to neutralize them. So this is an endogenous antioxidant system, which is pretty neat. But you might wonder, what is this reduced glutathione? What is this GSH? Well, what exactly does this represent? What's going on? Well, this reduced glutathione, this endogenous antioxidant, is simply this molecule, where this G represents this component, and this SH represents this Thous sulfur group, this sulfur and hydrogen thiol group. And really, all it is a glutamate, cysteine, and glycine linked together, where essentially we take a glutamate and react it with the cysteine, forming this gamma-glutamyl cysteine, which then we throw in a glycine to form this glutathione. So that's essentially what glutathione is. It's just 3 amino acids linked together. And this is what oxidized glutathione looks like, where we have these 2 G's and these 2 S's. Again, these G's just represent this component, and these 2 S's just literally represent 2 sulfurs covalently linked together. So this is oxidized glutathione. So remember, our electron transfer chain is constantly producing this superoxide free radical. And we know we have this superoxide dismutase, which can take this superoxide radical and convert it into hydrogen peroxide. However, this hydrogen peroxide can react with iron and other metals to form this toxic hydroxyl radical. So our cells are constantly going through this process producing these toxic hydroxyl radicals. So this is bad. This is not good. However, if we have strong regulation of these enzymes and strong regulation and upregulation of this peroxidase enzyme, instead of going through this pathway forming this toxic hydroxyl radical, we can go through this pathway where we take this hydrogen peroxide and convert it into this neutral water and oxygen gas. So if we want a strong antioxidant system, and if we want to minimize reactive oxygen species damage, we want to strongly upregulate this pathway while minimizing this pathway. So how do we do that? Well, first of all, we need to make sure we have the right minerals and cofactors to make sure these enzymes are functioning properly, which includes magnesium, copper, zinc, and selenium. So we need those minerals and cofactors to allow these enzymes to function properly. And if we have those minerals and cofactors, these enzymes are functioning so we can go through this good pathway. Also, we need a healthy diet. I really like curcumin and cruciferous vegetables like kale and broccoli and other cruciferous vegetables because they upregulate this transcription factor, this NRF2, which is a transcription factor. So it upregulates the expression of the genes that produce this peroxidase and this superoxidase mutase. So if you eat a good healthy diet, curcumin, cruciferous vegetables, and other good healthy foods, you can upregulate this pathway and upregulate the expression of these enzymes. So therefore you can maximize this pathway while minimizing this pathway. Also, what's important to realize is this particular enzyme, which allows this pathway to occur, requires glutathione. Glutathione has to be around so it can donate electrons to allow this pathway to work. So you want healthy levels of glutathione. So if you want high levels of glutathione, you need a lot of glycine. Because again, glycine is a component of this glutathione. And in fact, if you're low in glycine, your body is going to degrade glutathione to produce glycine. And also, I like alpha lipoic acid and N-acetylcysteine because they're proven to increase the levels of glutathione. And remember, it was that pentose phosphate pathway that produced that reduced glutathione. So therefore, it's important to have healthy levels of this pentose phosphate pathway to produce this reduced glutathione, to donate electrons and allow this enzyme to work and allow this pathway to occur in high levels. But also, if you want a healthy antioxidant system, you also need vitamins. So which vitamins? Well, again, remember this toxic process that's occurring with our phospholipids when they react with these reactive oxygen species? Well, we can stop this process if you have high levels of vitamins. For example, vitamin E and vitamin C. So how does this work? Well, again, remember the pentose phosphate pathway where we can take electrons from glucose and donate them to NADPH, which can then donate those electrons to glutathione, which can then donate those electrons to vitamin C, which can donate those electrons to vitamin E, which can donate those electrons to this particular intermediate, essentially stopping that chain reaction, forming neutralized fatty acids. So therefore, if you want to stop this toxic chain reaction that occurs with our phospholipids, you need high levels of vitamin E, vitamin C, and glutathione. So therefore, if you want a healthy endogenous antioxidant system to protect cell membrane integrity and to quench free radicals, you need a healthy diet with vitamin E and vitamin C. And again, those other cofactors we needed for a healthy antioxidant system. Also, if you eat a well-balanced diet, you'll ingest a lot of exogenous small molecule antioxidants. And you've probably seen in commercials, eat your antioxidants. Well, if you eat a well-balanced diet, you can ingest antioxidants. And these antioxidants can react with free radicals. So when they react with free radicals, they essentially donate electrons to these free radicals, neutralizing these free radicals. So it's important to eat a well-balanced diet, to eat a lot of exogenous antioxidants. And my favorite antioxidant is this EGCG, which is found in green tea. So I highly recommend reading about this EGCG and reading about all the incredible health benefits about this exogenous antioxidant. It is perhaps my favorite molecule in general. But generally speaking, this is how small molecule antioxidants work. So again, that's the conclusion of this video on free radical biochemistry.